am the President and CEO of Athera, and I want to welcome everybody to this breakfast event that we're having. The purpose of this event is really to talk about a multi-omics approach to personalized medicine. At Athera, we take a multi-omics approach to personalized medicine, and we really want to educate people on what we're doing and how we do it. So from there, I'll have the panel introduce themselves.
Hello, I'm Dr. Chip Petricoin. I'm a co-director for the Center for Applied Proteomics and Molecular Medicine at George Mason University and Chief Science Officer at Athera.
Hi, good morning. My name is Subha Mandaman. I direct the Innovation Center for Biomedical Informatics at Georgetown University. I'm also an associate professor working on cancer informatics research at the Lombardi Comprehensive Cancer Center, and I'm the Chief Data Bioinformatics Officer at Athera.
I'm Mike Bushman. I'm the GI oncologist. I'm at Georgetown University Lombardi Cancer Center. I'm director of our Phase 1 program, and I'm Chief Medical Officer for Athera.
So at this point, we just want to set the stage from each of their perspectives of what a multi-omics approach is and what's the latest developments in the field.
So my role in my academic job was to develop new technologies that go beyond a genomics-only approach to looking at proteins and whether or not they're expressed and whether or not they're activated and in use. When I was at the FDA for about 15 years, it dawned on me pretty quickly that proteins are very important. Most of the time, they're the drug target itself. Almost all of the targeted therapies and even targeted immunotherapies, checkpoint inhibitors, are targeting protein and protein activity, not genes. And many times the therapeutics themselves can be proteins, especially the biologics. And many biomarkers for diagnosing cancer, things like CA125, for example, these are proteins. So diagnostics oftentimes are underpinned by protein measurements. The drug targets for many of our targeted therapies are protein targets or even activated proteins. And sometimes the therapeutics themselves are proteins. So that led me into a field of looking at developing technologies for measuring proteins. But as we see with the exploding content of drugs and drug selection that are even FDA approved and off-label for a physician, this requires beyond, say, just measuring HER2 or measuring EGFR or measuring ER or AR. Now you want to be measuring all of those plus maybe 30 other things that may be in fact directly related to a biomarker that an FDA-approved therapy could be used for. So multi-omic analysis, multiplexed analysis becomes very important. And this leads then into a larger discussion of how much tissue does one need to get for being able to measure 50 things, and how do we actually do this from the clinic and control the pre-analytical variables and everything that goes with measuring not just a gene transcript level, not just measuring whether or not you have a HER2 mutation, but actually measuring it both at the genome, transcriptome, proteome, and maybe phosphoproteome level. And this is the challenge that Athera is taking on to provide the physician with that comprehensive, almost pathway-centered information.
Right, so my perspective is one of a data scientist. I've been playing with big data for over a decade now in the context of cancer research. So in preparation for this panel discussion today, I jotted down a few numbers just to sort of put things in perspective for us. And so before I sort of cite those big data numbers, which of course we all read about it constantly in journal articles and trade magazines, my team's goal is really to integrate a variety of data sets to drive better cancer therapies to drive health and healthcare. So the numbers I jotted down this morning, so if you look at Medline, for example, Medline has 21 million citations, 5,600 journals in 40 languages, and there are 27,000 mesh headings. So you all know what mesh headings are, so the medical standard headings. So these are just the headings, not the data. So you then associate data with these medical headings, and you've got big data in front of you. So So if you just look at Kaiser as a healthcare system, and they take care of 9 million lives, and they have approximately 44 terabytes of information. Terabytes of information, this is 10 to the 15, right? So this is just Kaiser. The world's sequencing capacity, right? So we've now been talking about lower cost of next generation sequencing. World sequencing capacity is 13 quadrillion DNA bases a year. This is just in 1 year, and that's 3 exabytes of data, so which is 10 to the power of 18. The way I explain this to my son is if you put this data on those 1.6 millimeter CD-ROM, well, first he asks me, what is a CD-ROM? I say, I don't know that. So if you put them and you stack them up, you can go from the Earth to the Moon and a quarter of the way back. This is 1 year's worth of DNA sequencing data, right? So, and there are 47,964 variants of unknown significance in the database of records called ClinVar, which is maintained by the National Library of Medicine. So this is the complexity of data that we're dealing with. So there's data from literature, data from clinical trials, data from sequencing that my team tries to make sense out of, right? So we develop methods to organize this data set, integrate this data set, add metadata to it so that we can support the bench scientists and the clinicians to generate better hypotheses for novel therapies.
So, my job as a clinician and somebody who directly sees patients on a regular basis is to distill all this information that's even overwhelming for me to something that the physician and the patient can actually utilize and take to the clinic and actually start to prescribe therapies or hopefully benefit patients. And there's really been an explosion of targeted therapies and biomarker development and trying to understand what is unique about each individual cancer patient. And if you take lung cancer as a perfect example, even though I'm a GI oncologist, I think the lung cancer has really developed broadly in this field, dramatically in this field. 10 years ago, lung cancer was really just one disease, and the median survival was about 8 to 10 months, and patients all got the same, regardless of the therapy they got, they all had the same outcome. And now here we are in 2015, and lung cancer is actually many, many different diseases, and a good 25% of patients have very dramatically activating mutations that, if appropriately targeted, have made humongous impact on their survival. I've got patients whose family members have lung cancer now, and if they happen to have an ALK rearrangement, if they happen to have an EGFR mutation, we're not talking about them, getting their affairs in order in 8 to 10 months. We're talking about them living 3, 4, 5 years based on these abnormalities. And again, there are multiple other slices of the pie in different diseases that we're seeing. In pancreatic cancer, we have the BRCA mutant and homologous recombination deficient patients who are really doing incredibly well with PARP inhibitor-based therapy and even just platinum-based therapy. In colon cancer, there was a great presentation last night, if you all went to it, that looked at the 5% of HER2 overexpressing colorectal cancer patients. In a refractory patient population, you had a 30% response rate with a combination of trastuzumab and lapatinib in what was called the Heracles trial. It's really showing that we're actually starting to subdivide these diseases into several different molecular categories, and doing so is making a dramatic impact on these patients. So our job is to take this multi-omic data, to run it through our tumor board, which includes disease-specific experts, a lung cancer specialist, a breast cancer specialist, etc., and to distill the information in a way that says, how would you treat that patient, and what combination therapies would you offer for that patient, either off-label, on-label, or actually what would be the perfect clinical trial for that patient.
There are some really practical issues of doing molecular profiling in the clinic today. And we'd like to ask Mike, what are some of those practical issues and how are you addressing them in your clinic today?
Well, so it's interesting actually. Before I really became involved with Prothera, I didn't do much profiling because it is logistically a pain in the butt. You have to get the tumor tissue, you have to requisition it from your pathology lab, you have to fill out forms. You don't have any way of sort of bird dogging or overseeing the quality assurance of what you're getting. And as we've learned, and certainly has become the way of thinking in a modern time, we really want to also get current tissue. We don't like to call it fresh tissue because that implies that it's non-formalin fixed tissue, but current tissue. And so to arrange a biopsy, to tell the interventional radiologist exactly what kind of tissue you need, and ultimately to get it sent off is a logistical headache. And when I have to do this for my patients at Georgetown, I have to spend quite a bit of time getting this all organized and filled out. And so Prothera has made it much easier to be able to do that, because basically the physician now is being asked to fill out one form and then it all just happens magically. And behind the scenes, the magic is that our team is doing a lot of that bird dogging and coordinating tissue collection and sending it out to the different companies and making sure that the information is coming back in. The other piece of it is the amount of tissue that Chip alluded to a few minutes ago. You know, I have a very good friend of mine who's an interventional radiologist and who I really kept pushing and pushing to get more and more tissue. And he said, Mike, you know, I was trained to get as little tissue as necessary to make a diagnosis. And now all of a sudden you're completely reversing that paradigm and you're asking me to get a lot of tissue for all this testing. And I said, yes, but that is really the changing tide. And so we need to help educate some of these proceduralists, whether it's interventional radiologists, surgeons, or even the GI docs who are now doing core biopsies from endoscopic ultrasound, to say we need a lot of tissue, and the more you do it, the more you'll realize that it can be safe for these patients.
And I can add, just from a sort of a data perspective in terms of all of it, so once the procedures are sorted out and the data is collected, you know, how do you then sort of integrate this information? So we're talking about genomic data, we're talking about protein data and phosphoprotein data that Chip mentioned. So we're developing a unique three-dimensional model that incorporates the multi-omic profile of the patient. The second dimension is the patient history. So what was the patient treated on? Is it a prior treated tumor? What did they get? Did they respond? And so on. And then the third is all the data and information from literature about this disease type. So if we're looking at metastatic pancreatic cancers, what's known? What are the on-label therapies? What are the off-label therapies? What are the guidelines that are available? And sort of incorporating all these three dimensions and making and ranking the therapeutic options for the patient. So we spend a lot of time thinking about, in a very thoughtful manner, how do we incorporate these detailed information that come out of the labs in order to make the best recommendations for our patients.
And I guess even to expand upon that a little bit, the logistical issue that we can provide tends to then homogenize the chaos of the pre-analytical variables that can exist in an uncontrolled state. So if these were just, 5,000 physicians all doing all this very difficult work, kind of with their own entropy, you'd never really be able to compare results from an academic standpoint, because you have so much variability in the way the tissue was cut, perhaps the pre-analytical variable, how long the tissue sat out on the bench before it was actually processed, et cetera. And by going through a common SOP that we've been able to develop, we can start to homogenize that. Now, it's still, you have to be practical. At the end of the day, it's a person processing the tissue. It doesn't happen by magic. There's no robots out there doing it. It's not an automated procedure. So, we're not myopic enough to think that even using a common SOP is going to solve all the problems. But it basically provides a launch point. And we're working with folks, say, at PanCan, for example, who we've built a great relationship with, that we can even work with members of their own scientific advisory board who are thought leaders themselves in many of these trials. And we can work with them early to develop best practices for collecting tissue that can become standard procedures. We can publish on these, we can compare, we do it in a very rigorous way. Because, as they say, tissue is the issue for many of these studies. And many pharmaceutical companies are basing their lives, their pharmaceutical companies' lives on the results from these trials. Geez, you'd hate to think about, missing an important biomarker because of how the tissue was collected. Again, one of the things we can provide is a standardized launch point for this that we can perhaps even in a year from now, 2 years from now, begin even to tighten that down. So we have a standard procedure, and we're constantly evolving whether or not this procedure provides the best. One of the things that we need to do, and we try to do, is because we believe in this multi-omic kind of comprehensive genomic, proteomic, transcriptomic, phosphoproteomic analyses to give all parts of the elephant, if you will, is using assays, CLIA assays, that are also best in class and are able to multi-class. So because we are not running the assays ourselves, it gives us the ability to stand above the process and choose best-in-class molecular profiling that each of these can be vetted by different metrics. One of the metrics that you can vet this is not just the turnaround time and the actionability of what they're providing, but also how much tissue do they need to be able to give you what you need, and how valid is that? What is their data? And then we can pick and choose from those. That in itself takes a lot of time for any single physician to keep up on. We provide that backdrop. And all of this is the ecosystem, I say, the ecology of providing back a personalized medicine service, a report that the physician can act on.
Thanks. So one of the questions I think that's out there is how significant are the genomic markers in really guiding treatment response? So we'd like to have Chip talk about his vision of incorporating genomics and incorporating other data into this.
But I think one of the things that we're finding from some of these trials is the prevalence of many of these genomic actionable markers is actually quite low. And that provides a conundrum. If you look at the custom trial, for example, where this is the lung cancer trial, that many of the markers they were looking at, say HER2 mutation or EGFR copy number variants, for example, were extremely low, so low in prevalence that there was no real way for you to stratify patients to those drugs had like 5 patients stratified to them, 7 patients stratified to them, hundreds and hundreds profiled. And this provides a pretty, I guess, depressing look at, how you would take a single kind of molecular profiling approach to, say, a basket trial or a bucket trial. My laboratory is involved in the I-SPY2 trial. We presented data here. We're actually the only molecular for neratinib in triple-negative breast cancer patients, which was interesting because a HER2-directed therapy was actually given to triple-negative breast cancer patients. The drug didn't graduate in that trial, but we found that if you measured phosphorylation of EGFR and HER2, the drug targets for neratinib, because it's a multi-kinase inhibitor, profound inhibitor, actually the path-CR rate blew away the graduating arm. And the graduating arm was HER2 positive, HR negative breast cancer patients that are only about 9%, whereas triple negatives are maybe 3 times the prevalence of that. Now, if you look at that population, that was found only by looking at phosphorylation of drug targets and asked, well, what was the prevalence of HER2 mutation? Would you use HER2 mutation? And the number of patients in the triple negative arm that had a HER2 mutation, 0. There were no patients that were HER2 mutated. In fact, in all patients looked at in I-SPY2, it's about the prevalence rate that Matt Ellis reported, about 1 to 2%. So that, to me, shows you already that, for example, you have a drug, we know it targets HER2, targets EGFR. By the way, EGFR copy number variants or mutation in that was 1. So we know that it can't be explained by a genomic approach to looking at the drug target. Because the prevalence is so low, but we know the response rate was higher than that. The PCR rate in the triple negative unselected population was 25%, 26%. Didn't surpass the control arm in an unselected population. However, they still had response rate. It was not just because of the HER2. Now, we looked in that phosphorylation, then their PCR rates were 70% in the biomarker positive population. So to start to focus us on, if we had a different part, again, of the lion and the elephant analogy, if we were looking at a different part of the elephant of this tumor, we may have been able to see a responder subset. And as I said, 50% of the patients were biomarker positive, 70% PCR rate of a 30% population. That number is much bigger than the unselected graduating arm by looking at a different marker. And this is what we're trying, and you don't know what you don't know going in. That's the point of looking at these biomarkers. And this is also why we want to take an approach. We look at the genomic analysis. We're not discounting that by any means. But then looking at these drug targets at a multi-omic level to pull in evidence that we can then act on, the physician can act on. And we want to be able to do this across both prospective clinical trials and for physicians who are treating heavily pretreated patients that have basically progressed off of standard of care. And we can provide a molecular rationale and a ranking system that allows them to make a decision is something that we're seeing academically as being very important. And I think there's some recent trials Mike was telling me about this morning that were announced here that actually even support this multi-omic analysis.
And just to go off of that comment, our ability to process patient tumors really does depend upon what's available out there in the retail market for diagnostic testing. And right now, the company that does the phosphoproteomic analysis just does breast cancer, but we're starting to see some intriguing potential benefits in other disease types. There's one patient that we came across with a pancreatic cancer whose genomic profile revealed a KRAS mutation and a P53 mutation, which really wasn't very actionable. But on a research basis, sort of post hoc, we actually had that patient's tumor profile for phosphoproteomic profile, and they had an active JAK-STAT pathway. And it really highlights the possibility, the potential for getting that patient either on a ruxolitinib-based therapy or, better yet, on one of the ruxolitinib trials that are ongoing right now. And perhaps that patient might be able to benefit more than would be otherwise expected because we're actually targeting a pathway that is acted by phosphoproteomics. So unfortunately, that was retrospective. But as these other disease types become available to test in real time, this may actually change how we move forward. And that phosphoproteomics should be available for breast cancer. It is available for breast cancer. It should be available for pancreatic cancer, for lung cancer, and for colorectal cancer. We think in the very near future.
And there's preclinical data that's really supporting this. And so if the National Cancer Institute put out a green challenge last year, which some of you may be familiar with. The goal of that study, of the challenge itself, was to look at breast cancer cell lines. This was a whole bunch of breast cancer cell lines where there was data that was available from DNA copy number, from gene expression data, from RPP and phosphoprotein data, methylation information. And the challenge was for international teams to look at all these data sets and see which ones actually better predict response to different drugs. So which cell lines were most sensitive to different drugs? Can you use all of these different data types to predict that? That was the challenge. And our team participated in that challenge, and we used a machine learning approach called Elastic Net, and we showed that the phosphoprotein data predicted the drug sensitivity ranking much higher than compared to other data types. The gene expression was over 20% accuracy in predicting drug sensitivity. This was published in Nature Methods last year, so if you're interested in looking at more details. So definitely there's academic and preclinical data that's beginning to support the multi-omic approach, and we're also seeing that in the patients that we are analyzing. We find that in about 40% of our cases, the ones that we processed at Prothera, that we actually have clinically actionable genomic mutations. What do you do for the other patients, right? So there we have to look at protein expression data, phosphoprotein data, and really see are there other options for these patients when all you have is TP53 and APC mutations. You can't do much with those. So I think there is sort of the T1, T2, T3, T4 data sets that are emerging for multi-omic support for therapy prediction.
Yeah, and to that point, data science and bioinformatics is playing a much larger role in personalized medicine than it has in the past. We'd like to super just to get her perspective on what role it is playing.
Well, I think, obviously this is self-serving, but I think that data science has a very critical role in personalized medicine. Just last week, O'Reilly published a book that I think the title was Navigating the Health Data Ecosystem, and they talk about the 6 C's, right? So with big data, you always talk about velocity, variety, volume, and all that, but it was interesting, the title actually talked about the 6 C's. And the 6 C's are complexity of the data. Of course, we know that this data is extremely complex, right? So if you compare the big data in biomedicine to, say, oceanography or astrophysics, There you have lots and lots of data, but you have same types of information repeating. Whereas here, you have heterogeneous types of information. So there's a lot of variety compared to oceanography information. So the complexity is a big deal. Second is computing. So a lot of the data, that Mike deals with on a day-to-day basis is not really available in a computable form because they're not intended to be computable, right? So they're for billing purposes, for taking care of the patient in front of you. So a lot of the data gets captured in unstructured form in clinical notes within electronic health records. So a lot of the data is not computable, and that's where sort of the gold nuggets lie, right? So we obviously want to mine this information that are in clinical charts. So then there's obviously the culture. So that's the third C. So they talk about how the culture of healthcare needs to change, and rightfully so, clinicians are risk averse, right? Because if they made a small mistake or if they try to take a risk and try to be innovative with their patient, it can affect somebody's life. So there's this extreme focus on evidence and how do you sort of incorporate evidence into your practice. And now we have digitized health, and everybody has their Fitbits, and patients are collecting data, and those data sets are being integrated with EHRs. Can the physicians readily use that information? Not yet, because there's not enough information to support use of that digitized health information in their day-to-day clinical care as it's approved by guidelines. So I think culture is a big issue. Commerce, this was actually a fascinating C for me when I was reading that book, was that investors want to see that there's at least $1 million or $2 million of revenue that's already generated in small businesses before they begin to invest in these innovative healthcare companies, because the payment models are crazy, right? So it's extremely complex, especially in the field of precision medicine. You have to navigate how do these reimbursements work, right? So if you look at the CPT codes, there are 2 CPT codes for 50 to 100 genes. Most of the molecular pathologists that I talk to say that you cannot use the CPT codes, so you get no reimbursement for those. And they have to use a combination of CPT codes while they're using these genomic testing. And there are a few other C's, but I'll stop there. So if you are interested, it's actually a free ebook, and it's a fascinating read, and they talk about the role of data science and big data and navigating data in the healthcare space. And I'm an optimist, and I like to think that if we can be smart about leveraging all this big data, I think we can only go upwards. I mean, we can do better. We can do better with multi-omics. We can do better with improving our clinical guidelines. We can do better with supporting patients with metastatic cancers who are progressing after first or second lines of therapy who have no options. And I think data can really be the solution. Yeah. My humble opinion.
So, reimbursement continues to be an issue in the molecular diagnostic world. And garnering reimbursement is relatively challenging. So we would like your guys' perspective on what the managed care is looking for from data and from the outcomes.
I think I'll take that. I think that one of the immediate challenges as you deal with the patient is, can I actually get access to that drug off label? And I think our experience with there has actually been very positive in that regard. There is definitely some regional dependence about who can get off-label therapy authorized. But we've had physicians tell us that they got our Prothera report, and to try and get authorization for a very off-label drug, they submitted our report. They didn't do any extra literature search or anything like that. They just submitted the Prothera report, and the insurance company acquiesced to give access to the drug. Now, that may not be true in every market, and every insurance company certainly is different. But the goal, ultimately, in the big picture, for all of the companies doing molecular diagnostic testing, and companies like ours that are definitely in this space, the ultimate goal is to link all of this testing to outcomes data. And that's why we put a very heavy emphasis on bugging the physicians to give us their records for the patients, to follow up with them on a regular basis, so that we actually can see whether the recommendations that we've made, if implemented, have actually changed the outcome for these patients. And in fact, we even are talking about how can we implement more, encourage more the physicians to actually follow up on our recommendations, so that we can really assess fairly whether we're changing the trajectory for these patients. If in a year we actually have 100 pancreatic cancer patients, and we find that 50 of them followed our recommendations and 50 didn't, and we actually see an impact in outcomes, then we're going to go to the insurance companies and say, we really are making a difference in these patients' lives when we incorporate multi-omic testing. And not to discount the possibility that we also potentially save the insurance company money by not letting patients get therapy that wouldn't have benefited them, actually. So we're trying to test this also in in the model of a formal clinical trial. It's not directly a Prothera effort, but certainly is overlapping with the Prothera efforts. So we were awarded, actually all of us at the table here, were awarded a PanCan ACR grant to do a phase 2 randomized trial in second-line pancreatic cancer therapy, where patients on first-line therapy get a biopsy, they get multi-omic profiling, including a lot more deeper science. Then a medical review panel would make a recommendation for the treatment for those patients, and then the patients get randomized to either follow that molecular tailored therapy recommendation or standard of care as determined by the treating physician. And the hope is, of course, that the patients who are on molecular tailored therapy will have a better outcome as measured in hard numbers, like in our case, progression-free survival, that we can actually improve the progression-free survival of patients based on their molecular tailored therapy recommendation.
I think there's some predicate for that. I think Mike hit the nail on the head in the sense that at the end of the day, the payers are looking for the same thing that all of us are looking for: true, rigorous evidence that this is changing the outcome. And we're hoping for things that are not something you need like a magnifying glass to see the difference in the Kaplan-Meier curves between the predicate. We're looking for big differences, for dramatic differences. And ultimately there goes back to the logistical issue, especially when you were doing this in a democratized way, where we want to give this access to all types of cancer patients, not just those that are delivering personalized medicine, not just to those patients that are able to make it to our comprehensive cancer centers, but maybe are treated at the community level, and being able to deliver to community oncologists the same level of molecular information as, say, an academic oncologist at one of these comprehensive cancer centers would be a great thing for cancer patients. Ultimately, the logistics of getting the outcome data become democratized in a negative way, in the sense that you have to follow these patients in clinics that are very busy, overwhelmed, have no staff to be paid to do this. And you want to get the outcomes data to support this, because at the end of the day, you need the outcomes data to do this. And one of the things, of course, that is said by the physicians is, what evidence do you have already? What evidence do you have already? We're trying to collect this. And as I said initially, there are some predicates seeping out there. We published a study in metastatic breast cancer from the SIDE-OUT trial about 2 years ago, where it was actually a multi-omic analysis was performed using protein, phosphoprotein, next-gen sequencing, and multiplex immunohistochemistry data. And what we used was the progression-free survival interval to see if the progression-free survival interval was increasing on molecular assay-directed therapies on patients who had progressed off of standard of care, or even third or fourth or fifth or sixth line. And we compared their PFS interval to the preceding PFS interval, something that Dan Van Hoff kind of made famous in his BISGROVE trial, to see if, because as you know, usually in that progressive setting, you're getting shortened PFS, not lengthened. And in that study, which we published, was that a small number of 25 women, but we had over half the women actually had an increased PFS of greater than 30%. Some of the women had an increased PFS of like 90%. And so this, was statistically powered in that number to be able to show a 30% increase. And we saw that. Now we're into a bigger trial to see if this holds up. And this is on a multi-omic study where the physicians, if you look back, were using the information both in a positive and negative way. When I say negative, meaning, wow, I'm glad we looked at that because I would have given that. And if I didn't have that, I would have given that. And it was again, it was the ecosystem of the multi-omic approach, which gave each physician at the molecular tumor board the decision to say, I'm going to give dasatinib to this breast cancer patient because it looks like SARC is activated. It looks like all these other things that I may have considered are not. And I will go ahead and see if BMS can give us this drug because of it, and for the outcome. Getting that outcome data is very, very important and requires logistics to get.
Thanks, Joe. So the last part of this is, how does the digital health really play a role in personalized medicine now. And Suba, we'd like you to kind of talk about that.
Yeah, so before I get into digital health, there are two sort of important aspects that I want to highlight. One is, and I think this relates to both what Chip and Mike were saying about, what is the evidence that is there that really, when we go and talk to clinicians about their service, they say, how many patients have you helped? How many pancreatic cancer patients have you actually helped with your multi-omic profiling? So we have an effort to really catalog the evidence that's available in literature and in clinical trials. We organize the data in multiple levels. So you can think of sort of level 1 evidence being from randomized controlled trials, but randomized controlled trials are not always available. When you look at protein markers, for example, so if we look at DNA repair proteins in terms of sensitivity or resistance to platinums or taxanes, and those are more level 3 type of evidence. So if you look at EGFR mutation and erlotinib, that's level 1 evidence. So we weight these different types of evidence in our ranking model before we say, yes, we're going to make this therapy recommendation. So I think that's one. Second is there's this national debate now about actionability of genomic variants. So the NHGRI, it's one of the NIH institutes, it's funding an effort called ClinGen. So that's why I was not able to be here on Thursday and Friday, because I was at that ClinGen conference where the discussion is about how do we classify these variants in terms of actionability, right? So is it just the driver mutations? If it is part of a pathway that is targetable, do we consider that actionable? If it is actionable through an on-label therapy or an off-label or an experimental therapy, where does that fall in the classification scheme? So there's this national debate about how do we sort of classify these variants. What is the evidence framework to assess what works and what doesn't work? And all this data that goes into this evidence are not always from randomized controlled trials. And I think as a country, we really don't have the resources to be doing large-scale randomized controlled trials. We've got to be smart about these interesting study designs, such as basket trials, for example, smaller patients, adaptive trials, MySpy. So how do we leverage data in a smart way rather than doing these 10,000 patient large-scale trial with a particular gene mutation? We're never going to be able to do that. So I think there's this concept of evidence that I wanted to highlight, which is such an important aspect of incorporating that into the individual patient's multi-omic profile. So talking about digital health, I can talk about digital health all day. It's my favorite topics, and I think there's a lot going on out there that we can leverage. So almost like a kid in the candy store here. So Apple in March just realized the research kit, right? So it's the open source platform that allows clinicians like Mike to be able to quickly deploy and collect data from patients. This could be outcomes data, this could be diet information, whether they're being compliant in terms of drugs that they're taking and so on. So all of that information can be readily collected using ResearchKit. There's a lot of development that's going on in the cloud computing space, and we work pretty closely with the Amazon. So a lot of the data, Amazon actually helps you put data in a secure and scalable fashion. You don't need large data centers to set up something so that you could collect data, which is terabytes of information. You could do that in a matter of 2 days. Spin it up, and these virtual servers could store and make that data available. One thing that is rapidly changing is we used to, 10 years ago, right, I'm really old, I color all my gray hair, I've been doing this for a long time. We used to take data to tools. We would take the data and bring them to the tools. We can't do that anymore because the data doesn't fit. It doesn't fit in any of the mode in which I can actually transfer data. So I've got to bring the tools to data, right? So you need this central location where you can keep data. These are called shared computing environments where clinicians, scientists, data scientists can come together. And that's where I think we're seeing a lot of revolution in the space through cloud computing, using Amazon infrastructure, Google, IBM, all of them are working towards these shared compute environments where you could bring tools to the data which is stored centrally. And of course, the digitized health and personal digital data collection is the mHealth initiative is going to be revolutionary. Of course, the super optimist that I am, I tend to think of this as great opportunities, but at the same time, you've also got to be cognizant about the contracts, right? So the business associated agreements, the HIPAA compliance, and the patient privacy, and all of this needs to be balanced with how we use and leverage this information. So would the genomic information be considered the 19th HIPAA identifier? It's a national debate. A lot of people are thinking about this. We have the 18 HIPAA identifiers, date of birth, the social security number. Should the genomic information be considered the 19th HIPAA identifier? I only need 75 polymorphisms from you to uniquely identify you in a database. I don't need anything else. Just 75 polymorphisms from an individual, blood-based, from constitutional DNA. So I think there's a lot of effort that's going on in terms of security security and privacy with large-scale compute infrastructures that really need to be balanced with the individualized information that we're collecting. So I think digital health is going to help, but one has to be very thoughtful about how we leverage them for our use cases. Stop it.
Thank you. Yeah, we're gonna have questions now. Okay.
I'm on here with the permanenting of the group. So a question, what is the cost, what is the turnaround time of your product?
So the cost of the Pathera service, in all honesty, depends upon who we're working with and what relationship we've built. But the cost of the testing is borne primarily by the insurance companies. So we are using testing companies that will have the capacity to bill insurance directly. We vet the companies, we only utilize companies who actually have the willingness to bill insurance and not balance bill the patients for the difference of what the insurance companies don't pay. And they've emphasized this repeatedly, and at least in 2015, that is certainly the current environment. It may change in the future, but that's certainly the current environment. And the cost of the insurance company is the cost of each one of those tests that the multi-omic testing is being done, and whatever they're willing to pay for the testing centers.
Turnaround time is, we aim from kind of consent, the patient-doctor interface with the doctor introduces this concept of personalized medicine analysis to the physician getting a report in about a month.
The biopsy.
From
The biopsy is about 30 days.
Yeah, there are definitely cases where, actually my own patients, where I talk to the patient, I consent the patients, and yet we wait a couple of months to actually do the biopsy. So from biopsy to report, we're waiting for about a month.
And that's for the full molecular analysis. So even in that context, we work with different entities where bits and pieces of the molecular profile can come in and go into that entity. Say if it's a clinical trial or if it's a specific focused effort on a group of patients. But for the physician kind of ordering it to get multi-omic analysis done and get a physician report, or their report, it's about a month. Now, we want to, most of that's driven by the turnaround time of the assay companies. And as they go down, our time goes down as well.
So actually, once we get the data from the assay companies, it's less than a week for our analysis. So the integration of multi-omics and for the medical review panel to look at it and produce a report is less than a week.
Right, so practically speaking, I mean, it's great, but turnaround time, I think that could interfere with the quality of care. Because by the time, when we look at cycles, which are all familiar and technically sophisticated, when you look at cycle time, this is one step among dozens that patients are going to be involved in, and one step takes a month.
Yeah,
It's very
it is.
concerning.
Yeah, it is. There are a number of things that we can do to shorten the time from an operational perspective, and we're working on those internally. But what we can't control is the amount of time the testing laboratories take to do that. And from them getting it back to us, typically it's between 10 and 14 days. We can reduce time on the back end, but you're still going to look at about 3 weeks turnaround time. And I don't think that from an industry perspective, you're going to see as actionable a report as we deliver to the patients in a shorter amount of time.
And it's definitely taught us how to utilize these tests. So we actually don't want to sell to anybody that we can actually decide what therapy they need for a newly diagnosed pancreatic cancer patient. Because that patient needs to start on therapy. But a patient who's actively on frontline therapy can go through the process of getting biopsy and has enough time to get that report 20, 30 days later. It also gives us the time then, and also if the report says you really need some sort of off-label therapy or some really ideally suited clinical trial, to get that patient on that trial. So we dissuade physicians from using our service when they realize that they need to treat that patient tomorrow.
Thank you.
You talked about using the data prospectively, and that's what the promise of the data is. But what about taking the data and look at it retrospectively? I mean, we think that our first and second line therapies are set in stone, and God sent us the message that that is it. But in reality, those are not personalized therapies. And if we take this information and look back and see, is the PFS of 9 months that I was expecting in this genomic profile, is it really 9 months, or was that PFS 3 months? I think that's a data that's currently available and can be leveraged.
And I agree, and I think one of our efforts definitely is to be collecting the data and progression-free survival for the patient prior to when we did the analysis of the therapy that they were on, and then of course moving forward the outcomes data, and to be able to compare the two. So yeah, as our data sets grow, we'll be able to get an assessment of, based on their biomarker profile, what were their progression-free survivals on the therapy leading up to the profile and then following after the profile.
And we also actually collect the Karnofsky score, so the metric for how the patient was doing prior to getting that first or second line of therapy, so that we know that, was it because the patient was doing poorly that they didn't respond well, or, were there other covariates?
It's all about outcomes, it's all about the data.
Yeah, I think this is hopefully one of the great opportunities with some of these larger efforts. Again, with our partnership with PanCan, the Pancreatic Cancer Alliance, being able to look at, what hopefully will be the largest molecular profile of pancreatic cancer that has existed, deep multi-omic analyses, far surpassing TCGA and some of these efforts. We'll be able to look back at the molecular architecture of pancreatic cancer from our retrospectively, and that can also help us inform you. We can distill out some of these outcome relationships on the different therapies, because it's the physician's choice. The data that we collect goes through a molecular tumor board, also a virtual tumor board. So the physician actually gets a report that's been vetted by peer oncologists. It's not just a bunch of PhD molecular biologists and informaticians saying this is what you should do. It basically is very much a sterile molecular analysis that then goes into a molecular tumor board. But then it's the physician's choice, right? It's always the physician's choice to treat. We're not practicing medicine. So one of the first things we wanted to collect was, is this helpful? Is the therapy ranking that we give, do you use that? Is that helpful to you? And really, vast majority of the time, it was. Did the physician use the ranking to make a decision? Was the first ranked therapy the one that they actually chose, or was it the last ranked therapy? Because we put in all sorts of, it's all molecularly driven. So clinical trials that are accruing, that are molecularly rationalized based on the patient's profile are put in there. That are in geographic proximity to the patient are active and accruing. Often on-label FDA-approved therapeutics are all on there. Depending what the physician decides to do, it could be that none of these things they decide to do. So we first asked is, what's on here, what you used? Yes. Okay, that's good, check that box. Was it useful? Yes, check that box. But then it becomes, okay, well, how long did the patient live or progress based on that therapy? What did you give? And then how did that compare to the molecular profile of the other 100 patients that shared that same profile out of the 1,000 pancreatic cancer patients? And did that make a difference in the OS and PFS? And then what were the molecular correlates, biomarker correlates for outcome to, say, patients that were treated with this specific therapy? Because now you're looking at a mass of patients, 1,000 pancreatic cancer patients. And that's the hopeful opportunity that we want to have for all of our different tumors. And then we can start to look across tumor types so that things like, bucket type trials could become maybe more informed. Say, hey, looking at phosphorylation of HER2 across patients with melanoma or soft tissue sarcomas or breast cancer, all were basically informed by phosphorylation. And we have that kind of data to triangulate. That's the ultimate kind of goal as a company we have, that can publish on this and show the support for it.
And it's also intent to treat, right? So we also collect information from the physician, even if they didn't put the patient the very next day after seeing the report on one of our recommended therapies, did it actually influence their thought process in terms of designing the treatment plan? So it's the intent to treat is also some information that we capture. So we want to put the best data in the hands of the physicians, whether how they use it or not, it's their decision.
The three worst letters most frustrating to medical oncologists are QNS. And are you guys planning on working at all with any of the liquid biopsy companies? I mean, it seems to be growing, like weeds out there. And
So I'll try and in the interest of time answer that question fairly briefly. First of all, again, getting to the logistics of testing, you know, I think a physician doesn't appreciate the degree to which we oversee the process of testing. So there are times, not infrequently, where we send out a sample and it's not sufficient. And yet, because we've collected the sample and we have additional sample in the storage, we can go back and request a different sample. And there have been even times where we've had to go back and request a surgical sample to be able to get something that's useful. So that multi-layered processing of making sure we get enough tissue has led to about an 80 to 85% rate of having enough tissue to do the full multi-omic profiling. We certainly are actively talking to some of the liquid biopsy companies to see where do they fit in, and that's part of the benefit of it there also, is that we're not linked to any one company, but we're allowed to sort of stay above the fray and kind of delve into their logistics and say, you know, do we really want to incorporate the liquid biopsy into our SOPs? And we just had a conversation just this morning about this, about where this might fit in. So the answer to liquid biopsies is we think that we're going to incorporate it, but we really want to be sure about when and where we incorporate it. It's probably not going to be in every single patient.
I have a couple of questions. Follow up on that. How much tissue do you need? And my second question is about quality control with your members, because for one test, the p-value is 0.05 is good enough, but when you add multiple tests, even though each individual test is 5% tolerance, we add them all up globally, or becomes 20%. So you push the vendors to get their tolerance a little bit lower, in order to get global p-value of 0.05. You understand what I'm question?
I'll take the question about how much. So we do work with core needle biopsies primarily. Our SOP asks that the interventional radiologist, and usually it's interventional radiology, so it asks that the proceduralist gets 4 to 6 decent core needle biopsies from a metastatic tumor deposit, whatever's safe and most accessible. As far as multi-omic testing,
So that's how many cc's of tissue?
so core needle biopsy is typically half a centimeter to a centimeter in length, ideally, each core, and the diameter of a typical sort of 19 gauge core is a little less than a millimeter. So I couldn't tell you the cc's.
Yeah, I mean, so if the question is, what's the tolerance of tumor content, or do you mean more the false discovery rate in the context of
False positive or false negative.
Yeah.
Because, you know, each individual has, you set the tolerance at 5%, I think that's good enough, but when you like globally, like all 4 the tolerance becomes,
Right.
The error becomes accurate.
Right. So one of the things that we're looking for in the context of multi-omic analysis is not to triangulate disparate data together for saying, well, this patient was high phospho-HER2 and low topo-A1 and MET positive, and therefore here's a clinical trial that's combining a HER2 and MET inhibitor together. We're actually looking for evidence of synergy, where we're telling more of a pathway story. Where if we see a PIK3CA mutation, phosphorylation of AKT, an mTOR mutation, that becomes a ranking of a higher level of molecular detail of something going on with pathway level. There's a pathway story emerging. There actually is data dependency, where you actually do not have a Bonferroni correction there, because the data is dependent on each other in the sense that they're biochemically linked. And this is the kind of way that we're trying to use a multi-omic approach, is actually trying to find synergistic information. Because the challenge, of course, is none of us know what the true driver is in any single individual patient's tumor. We can infer, but we don't know. I mean, as Mike said, many of our pancreatic cancer patients are P53 and KRAS mutant. Great, you told me now that this is a pancreatic cancer tumor. You told me nothing about how to treat the patient. And so we know, until we find good RAS drugs as a potential undruggable target, that we can't use that information to really select therapy. You're not going to get giving a MEK inhibitor, forget it, it doesn't work. So what we're looking for is molecular data that builds on a pathway story. And so we're not using it as just little bits of constellation of different data sets, but each one has its own variability, and you're combining variabilities to give you just a big, high CV. We're trying to find evidence of a linkage.
So, just very briefly to add to that, I mean, you could sort of do integration at two levels, right? One is at the statistical level, where you get all the raw data, and that's where I think the point that you're raising in terms of tolerance and everything, and how do you sort of integrate data from these multi-data type platforms at the statistical level. Then there's the level of the pathways or the scientific integration. So we're more targeting at the second level, because the assay companies know their platforms best, and if we find issues, we're constantly talking to them to improve their platforms. But once we get the data, we trust their calls today, and being a platform agnostic company, it allows us to vet whether the platforms are high quality, is the data in computable form and whatnot. So we do that. But then the story we're trying to tell is more at the scientific integration level. So we're not actually taking raw data from Foundation, from CARES, and from PGDX and trying to do our own statistical integration. That's not something we're doing right now.
Are there any other questions? All right, well, thank you all for attending today. And the panelists will be available down there for questions.
Thank you.